In this video, we will discuss about HiFi Cas9 system comparison with the Sotorasib, which is a FDA approved drug that target this KRAS-G12C mutations. So before going into the results, let us understand the difference between the HiFi Cas9 system that the researchers has developed and the Sotorasib that is a FDA approved drug. So what it does is it will make the KRAS-G12C proteins that are being translated inactive. However, HiFi Cas9 system, it will inhibit the translation or transcription of the KRAS mutations, and this HiFi Cas9 system not just target KRAS-G12C mutations, but it will also target KRAS-G12D mutations, and these two mutations are commonly found in the cancer cells. So in the panel A, what they did is they checked the combined efficiencies. In this case, the experimental plan is that first they transduce the cells with adeno-associated virus control and adeno-associated virus with HiFi Cas9 24 hours before they inject Sotorasib in both the conditions. And what they are able to see that in the case where HiFi Cas9 system is used along with the Sotorasib, they are able to see a decrease in the decrease in the cell viability, but that decrease is not statistically significant compared to the case where there is no Sotorasib is used. The next, they plan to use they plan to do this experiment to check the amount of colonies that they get, and this time they use three different conditions. First one is the adeno-associated virus alone, and second one is the adeno-associated virus with Sotorasib, and third one is this control. Third one is let me again repeat the conditions. First one is their own design system, adeno-associated virus with HiFi Cas9 system. Second one is the Sotorasib without adeno-associated virus HiFi Cas9 system, and the third one is the combinations of both adeno-associated virus and Sotorasib. And and the combinations, all these combinations are shown in this panel B. The quantifications of these results in terms of the cell viability with respect to the adeno control, that is adeno-associated virus without any HiFi Cas9 system, is been shown or plotted in the panel C. And in this case, this TP181, TP79, TP60, all this represents the different patients derived xenograft samples. And so what they are able to see is that for the TP181 they do not see any significant difference, but for this TP79 they can see a significant difference with this adeno-associated virus with HiFi Cas9 system alone compared to the case of this Sotorasib or the combined. In the combined case also has a statistical significance compared to the Sotorasib, but they are non-significant compared to the adeno-associated virus with HiFi Cas9 system. And for the TP60 also, in that case they are able to see a significant decrease. However, the decrease in the case of adeno-associated virus HiFi Cas9 system and the combined is non-significant still. So the TP79 shows a results where the HiFi Cas9 system can works better than Sotorasib, and the reasons that they cited is because the mechanisms is different. And the immunofluorescence images are also shown here, where we can see that for the case of TP181 there is not much significant difference, but for the case of TP79, for the HiFi Cas9 system, we can see a decrease in the signals, GFP signals, and for the control this TP60 we also see some slight decrease in the signals. After that, validating that the HiFi Cas9 system works comparable works with the comparable efficiencies to compare to the Sotorasib. They also wanted to see how it will their system will affect or react behave to the cells that are already resistant to the Sotorasib. So in this case, they take the Sotorasib resistant cells, and they can see that when they in the panel E, we can see that when they use the 2D culture model, panel E is about 2D culture model, they do not see a significant difference in the cell viability. But when they use the 3D culture model, they can see a significant difference in the cell viability to up to 60%. And this cell viability is related to the tumors. So they can see a significant decrease in the tumor cells to about 60% when they use the 3D model. And so this indicates that their HiFi Cas9 system targeting KRAS mutations seems to work better than the Sotorasib on the Sotorasib resistant cells, or in other terms, it works better than the Sotorasib targeting KRAS-G12C mutations FDA approved drug. And the immunofluorescence images are also shown in the panel G, where we can see that a decrease in the cell viability for the H23, which is the control, and also we can see some decrease for resistant cells, H358 resistant cells 2 and H350 resistant cells 1. And here we can see the results that for the case of this H23, the results are statistically significant, but for the resistant cells, although we do not see a statistically significant results, but for the individual cells, if they look, they can see a decrease in the some of the cases. So this is what they finally conclude, that how their HiFi Cas9 system works at least comparable, and to some extent even better compared to the already FDA approved Sotorasib targeting KRAS-G12C mutations drug in the and this drug is not only approved by FDA, but also approved by the European uh, drug committee. So I was not able to recall that committee name. That's all for this video. If you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. Thank you for your time.